Active transport, vesicular transport. Vesicular transport is the active movement of substances across the cell membrane via a vesicle. In exocytosis, materials in the intracellular fluid are engulfed by a vesicle, which then secretes or empties the contents of the vesicle into the extracellular fluid. In endocytosis, materials from the extracellular fluid are engulfed by a vesicle, which then empties its contents in the intracellular fluid. In exocytosis, materials in the intracellular fluid are engulfed by a vesicle, which then secretes or empties the contents of the vesicle into the extracellular fluid. First, a vesicle containing materials to be transported approaches the plasma membrane, guided by the cytoskeleton. The vesicle then fuses with the plasma membrane and empties its contents into the extracellular space. In endocytosis, materials in the extracellular fluid are engulfed by a vesicle, which then empties its contents into the intracellular fluid. First, molecules approach the plasma membrane from the extracellular side. The plasma membrane invaginates to take in the molecules, forming a vesicle in the intracellular space. Phagocytosis is a special type of endocytosis in which larger particles are engulfed by the cell. For example, bacteria are consumed by white blood cells in this way. First, the plasma membrane invaginates to take in the particle, forming a vesicle in the intracellular space. Soon after formation of the vesicle, a lysosome joins the vesicle, emptying its digestive enzymes into the vesicle to break down the particle. Pinocytosis is a special type of endocytosis in which the cell takes in extracellular fluid. Typically, the fluid taken in by pinocytosis is rich in nutrients. The plasma membrane invaginates to take in the fluid, forming a vesicle in the intracellular space. In receptor-mediated endocytosis, molecules first approach a specialized area of the plasma membrane equipped with cell surface receptor proteins. Only molecules matching the shape of the receptors will bind to the receptors. First, the area of the plasma membrane where molecules are bound to receptors invaginates, creating a vesicle inside the cell. Then, new cell surface receptor proteins are manufactured by the cell to replace those used up in vesicle formation.